means to be free of a burden. And the immune system works by a learning process. Two steps. One, recognition. You must recognize self versus non-self. That's the first thing. Of course, recognizing self means don't react to that, react to non-self. And so if you have the ability to recognize something has invaded your body, you follow with step 2, which are the destructive forces that would follow step 1. Destructive forces that follow recognition. Of non-self, whatever the non-self is, a pathogen, a germ, bacteria, and so what you see on the slide there are what are categorized as non-specific or specific defenses. A defense that is called non-specific or sometimes they say innate. Non-specific or innate defenses defend against all things that are non-self, anything that's not from your body. So then specific defenses they require this adaptation, and they defend against a particular non-self bug. So specific defenses defend against a particular bug. And it's great to fight out that particular bug, but not any other one. It kind of adapts to this bug. Adapts to a bug. Now, the lines of defense, well, the first line of defense is just don't even let it in your body. Just keep it out, right? So first line of defense. Those are the membrane barriers. Keep things outside the body. Now the membrane barriers include, well, skin, that's the best one, keep it out. The largest organ of the body is the best membrane. It's a dry cutaneous membrane. You also have mucous membranes like that line your gut, line your throat. Mucous membranes. I also listed stomach acid, good old HCl. That's secreted by the stomach mucosa. And that the acidity of the stomach will keep a lot of things out of your system. However, if things actually do get inside the body, you have your second line of defense, which is your mobile army of phagocytes circulating the blood. Mobile army of phagocytes. Cells that execute phagocytosis. That's one of the best. That's one of the best ways to, I guess, murder the pathogens inside your body. Now, I list some there. Macrophages, which are derived from monocytes, right? So macrophages, eosinophils, dendritic cells. So those are some of the phagocytes. Macrophage, eosinophil, dendritic cell. Maybe I'll add neutrophil. So these phagocytes, they roam around the body, they're inside the body, they're just looking for pathogens, and they have the ability to recognize all things that are non-self. For example, the macrophage has this cell receptor called the Scavenger Receptor. I like that name. The Scavenger Receptor. Right? You just look at it, scavenging all the tissues to keep things sterile, looking for anything that's non-self. Okay. I also have listed under second line of defense interferons and complement, which are also a part of non-specific defenses. But I got slides on those. We'll get to that. The third line of defense are your specific defenses, where I erased it, but it's where I said you adapt to a particular bug. Third line. That's the B and T lymphs. You know, the B and T cells. Sometimes people say lymphs, they mean cells. And I'll talk a lot about these. Antigens are required. This requires exposure. Some way, shape, or form. Exposure. Well, to antigens, which I frequently abbreviate AGs. So in this lecture, AG is antigen. What's AB? Antibody. Terms you're familiar with because we talked about blood typing. Well, requires exposure. Then you can recognize, adapt, mount a response in that order. Recognize. Adapt / respond. I mean, there's different ways you can say it. I mean, I'm going to spell it all out for you. But we'll talk a lot about the B and T cells. We'll talk about them mostly, actually. This third line defense. It's the best one. Okay. It really does confer immunity in a way that if you ever get that bug again, you won't get sick. Okay, so I already covered this. Just to remind you, we did talk about white blood cells before in terms of how they form in the bone marrow. And remember we went through this slide? I just kind of showed you how they developed from different colonies in the bone marrow. I don't want to rehash that, but it's ground covered, and we went through how to identify them on a blood smear slide, and we went through the basic functions of them. So I'm just going to skip this. Ground cover. I think I'm just going to delete them for the next semester because I already covered them in a previous lecture. This part I didn't cover. This is teaching students the generalized, non-specific response of chemotaxis, interferons, and complement. Chemotaxis is a term that refers to cells migrating to an infection, and the non-specific defenses, they're not distinguishing one threat from another. And that phagocytic army of cells, they all participate in this chemotactic inflammatory response. Step one, vasodilate the area. Let blood rush in. That will attract leukocytes via chemotaxis. And here's a picture of that. The inflammatory response, part of your non-specific defenses. Step on a nail. Are you infected? Well, you see that white pus? You're infected. What's the white pus? White blood cells. They're attracted to the area of infection. And this slide has what's nicely outlined, but they leave some gaps. I'm going to fill the gaps in. So, the inflammatory response. Inflammation. We all know what that is. Redness, swelling, right? May be accompanied with the white pus if there's an infection. Now, certainly, in this example, we want the infection to be there, those green capsules. But then you see the damaged tissue cells secreting the blue molecules of chemoattractant proteins. Okay? So the first thing is, all right, well, we have an infection here. We need to recruit leukocytes to this area. That's what inflammation is. Recruitment of leukocytes. In this case, that's a neutrophil. Recruitment of leukocytes to the infected area. All right, so the first step they list is leukocytosis. This is just like get in the game. You can't stay in the bone marrow. You're not going to do anything there. You have to enter the circulation. So leukocytosis, step 1. Neutrophil enters bloodstream. It circulates. It's looking for things to do, basically. All right, so what's going on here is, one of the gaps is they don't tell you that in this infected area, the endothelial cells of the blood vessel will present adhesion molecules. Endothelial. Remember what we're talking about here? What tunic are those cells in? TI, right? Tunica intima. I'll put in parentheses of infected area. They're stimulated to produce the proteins for adhesion molecules. Okay, the surface is sticky, like Velcro. Now the adhesion molecules, what they'll do is when the right kind of cell comes on by, they'll roll. See these arrows? It's rolling and slowing down, stop. Okay? So neutrophil. It rolls, slows, stop. Then, well, the next step, number 2, margination. I call that adhesion. I'm not going to use margination. I always call that cell adhesion. Neutrophil adheres to the vessel wall. I mean, it's stuck on the inside. It has to get to the outside. It has to enter the connective tissue to do its job. And so that's the job of, well, they say diapedesis. I mean, what's happening is it's catching a whiff of these chemoattractant cells, which provide a trail, and it just kind of follows the trail, and it's strong enough to suck it through the vessel wall. Call that diapedesis. Diapedesis. Neutrophil. Well, this word means diped. Di means dissect, apart. Ped means foot, jump across. Neutrophil jumps across squeeze, if you will. Vessel wall into, well, the tissues of the infected area. Infected tissues. What they're not telling you is it's following that chemotactic trail of the chemoattractant proteins. So I'll just put it's following a chemotaxis trail. And that's what they list as the last step. Chemotaxis, follow the trail. Just sniff it out. I'll just repeat it. How about I just go like this? That is step 4. Okay, I don't want to write it again. But that's important, that chemoattractant protein, it was strong enough to suck it through the wall. And I used to do this in the lab when I was a grad student. I could just take a well, fill it with chemoattractant protein, put a paper membrane on top of that was porous, put white blood cells on top of the paper, and it would get sucked to the bottom of the well. They're attracted to those proteins. So chemotaxis is how they do that. And once they get to the bacterium, they execute phagocytosis. And that's the job. Kill that thing before it has a chance to spread through your body. How do you know it's spread? Maybe you get a fever a couple hours later. That's not good. You better go somewhere. Okay. I remember one time I was cleaning the toilet. Some water splashed in my eye. Oh no. I remember a short time later, my eye started throbbing. I go, oh no. But it stopped there. So I was like, hey, I was fine. But you know, that's what our immune system does. We don't know what's happening, but we know how we feel. There's a picture of what happens. The white blood cells phagocytizing bacterium. One mechanism against viruses are interferons. They interfere with viral replication. Interferons, well, these are molecules that, quote unquote, interfere with viral replication or reproduction, however you want to think of it. That's the key, because what viruses do is they hijack cells by inserting their own genetic material into the host cell's DNA. So the viral cells I don't know if they're cells, let's just call them virons. Their genetic material is RNA, but they contain a reverse transcriptase where you can go in reverse. You can go backwards from RNA back to DNA. Have a reverse transcriptase. So you go from RNA, this is the virus RNA, to DNA, virus DNA, and then insert into the host cell DNA. Insert this into host cell DNA. That's what you see in host cell 1, okay, on the figure. it's infected. And someone won the Nobel Prize for this right here. And they realized there's an enzyme where you can go in reverse, because before then, in the 70s, they didn't know that. It only went one way. That's important, because this is how viruses work. Now, what the infected cell does as a defense mechanism, not for itself, but for other uninfected cells, they'll release the interferon molecules after it's been infected. So keep track of the cells. Host cell 1. Infected. And as a result, in response, secretes interferons. But what host cell 2 does, it binds the interferons. It responds to the interferons by producing antiviral proteins. I'll say host cell 2 not yet infected. In response to interferons from host cell 1, produces antiviral proteins to block viral replication. So this cell is protected. Host cell 2 protected. Is host cell 1 protected? Yes or no?
No.
Nope, it's already been infected. But now you're stopping the thing in its tracks, and eventually you'll get better. It may take a couple weeks. What happens if you can't stop the replication? You can die from a viral infection, and many people do every year. There has to be something in your body that can kill or block the infection. Your body will always mount an immune response to whatever virus, and if you survive, it worked. Now, there's another component of the non-specific defenses called complement, shown on this slide. So there's a cascade of things that occur. It gets kicked off at the top there. Let me see. Has anyone taken micro yet?
Yes.
Have you talked about this stuff? Maybe? Yeah, it should be covered in more detail in micro. That's their value tricks. But we covered here too as part of the human physiology. All right, so I like the name complement because complement, that's what it does. It complements both specific and non-specific defenses. Complements both specific and non-specific defenses. That's what this slide is showing you. And how the pathway is initiated, the activation of complement. Let's just learn about the classical pathway that triggers complement. It's triggered by, well, the classical pathway is activated when antibodies coat the target pathogen or cell. Classical pathway. ABs, antibodies, co-pathogen. The fancy name for that is opsonization. So say you have pathogen bug. It's a bacteria or something, and it's got all these little surface antigens on it. And you have, let's say you have a particular clone of antibody that recognizes it. Now the antibodies are part of your specific defenses, mind you, but I'll get to that. So let's say that these blue antibodies can bind that pathogen. And you just coat the surface. Coat the thing. The thing about antibodies is I don't want to get too much into the structure. These immunoglobulins, or antibodies, as I like to call them. This fourth region is the specific region, specific for these little antigens on the pathogens. So fourth region, specific. The tail is non-specific. It's a non-specific region. And what happens is the tail region will just, bump into components of complement like C3b on the slide. C3b. Okay, so the tail bumps into it, and what it does is when this kind of like goes to the pathogen, it fixes complement to the pathogen. So let me draw a C3b here. So bring this to the party. And well, they call that complement fixation when that happens. Complement fixation. That's a good thing. Let me read what it says on the slide there. When you do that, you go to pathogen, complement fixation, you're enhancing the chance that this thing, this pathogen, will be phagocytized with this complement fixation. So I'll write that somewhere. You're increasing the chance for phagocytosis. This is the destruction. The coating is not. It's just kind of holding it down. But the phagocytosis is the kill part. Anyway, I always say, the antibodies, they're the accessories to murder. They're not the murderers. But you just kind of hold it down and wait for your army of phagocytes to come along and destroy it. So that's opsonization. That's how you complement the specific defenses. How you complement non-specific defenses, I won't say much. You just enhance inflammation. You increase the histamine release by the basophils. So you enhance non-specific defenses, just increase inflammation by increasing histamine release. By basophils. So let's call that, if you call opsonization number 1, call this enhancement of inflammation number 2. And the third thing that complement does, they execute what's called MAC attack. I'm not going to write it out for you, just remember it stands for membrane attack complex. Membrane attack complex. You're just punching a hole in that thing. And that's what they're showing you here when you assemble all of these complement molecules. I won't make you memorize them. Just remember that MAC stands for membrane attack complex. You take a bunch of these, arrange them in a pore-like structure, you allow lysis to occur. MAC attack facilitates lysis. And that's another way you can kill. Facilitates lysis. The thing will burst because water and other things will rush in. So the destructive forces are the lysis and the phagocytosis. Let's complement. So I want to shift gears and talk about the specific or adaptive defenses, which means we'll get into the B and T cell thing. Now the B and T cells are the lymphocytes. And what we have learned from past research is the study of the mice models that have the, well, remember I mentioned about the inbred strains, right? So you get the homogeneous genetic strains of mice. Well, anyways, what they do is they just, it's easy to understand. You inject animal with an antigen, you're provoking an immune response. And you got to do something to the animal so that there is no immune response. So they just irradiate the animal. High energy waves, they just kill cell function. And so when they inject with the antigen, the normal immune response is gone. So that's your model. Now you can say, well, which cells, which were eradicated in terms of function, which conferred the immune response? So what you do is take an animal that's been irradiated, no cell function, inject it with the antigen, inject it with some other cell, just line them up. How about connective tissue cells? Other cells, and if there's no immune response, what you say is, these cells have no immune function. But if you then inject with lymphocytes, and they respond to the antigen of the irradiated animal, and you restore the immune response, similar to the normal mouse, which has not been irradiated, you can say it's the lymphocytes that provide the immune function. And so that's what we're talking about here. The specific defenses require exposure. They respond to a particular bug, a particular bacteria, but they'll be ineffective against others. So that exposure, you live in the world, you get sick from well, your kids or your classmate or whatever. You're out in the world and someone sneezes on you, right? Or deliberate. This way, you get the memory cell, but you don't get sick. That's a vaccination. They give you some kind of antigen or attenuated form or some inactive form of the bug or virus. So that way you're not supposed to get sick. So specific defenses are lymphocyte dependent. The B and T lymphs are the two main categories, but there are subpopulations for each. So I have to define those. Well, just to give you an overview, the B lymphs, those are the antibody secreting cells. Just remember, B lymphs, ABs. I think they're called B cells because they started studying them from the bursa of birds, but you don't need to know that. We just call them B lymphs. Now, let's say, for example, you got some antigen in your circulation. Some non-self infection. I'll call it AG for antigen. That's the particular bug you got to get rid of. Let's say you have a particular clone. Let me draw a couple of clones. Let's say you have this blue lymphocyte. That's what they look like under the microscope, correct? Big, large, round nuke, a little room of cytoplasm. What you should know is that they have these membrane-bound ABs. That act as receptors. Okay. But you have many clones of these. Let me draw another one. You have a red one with membrane-bound AB. It's a different clone. So I guess, well, I drew a red one and a red one. So let's say that this clone is specific for that bug. Not the blue one, this one. So it can bind it, and that can trigger a reaction, okay? And that clone will be expanded and proliferated. So it'll just, undergo cell division. We'll draw a couple of them. Expand. And maybe this, these will further expand. Maybe this will become a plasma cell. These larger cells that have more cell machinery in the cytoplasm. Maybe there's an ER, maybe there's a Golgi apparatus. I'll just draw more cell organelles in there. So this is your antibody factory. Okay, I mean the original cell is not. There's no cytoplasm. But once you're activated, you increase the cytoplasm and you become this antibody factory, and you start secreting the antibodies. They're not just membrane bound. They're in circulation, and they can do their job separate from the cell. And the job of the plasma cell is to increase the number of antibodies for the bloodstream. On a drop of blood, you usually, for our slides, you usually don't see them because they're usually in the connective tissues, the plasma cells. Plasma. Remember that. The antibody factories. Maybe this cell persists as a memory B cell. Okay, there's a difference between the memory B cell and the one it came from. The one it came from was the original, but the memory B cell, they may persist for years, maybe decades. And if you ever get this bug again, you'll mount a faster, stronger response. You probably won't even get sick. For the T cells, there's also many subpopulations. TH. T stands for thymus. That's where you have the thymus education, the 2 rounds of selection. Well, anyways, H stands for helper. So the helper T cells are also called the CD4 cells. So if I ever use the term CD4 cell, same thing as helper T. The CD4s actually become the helper T cells. They're really important. They kind of like kick off the entire immune response. I would say they're the most important. Okay. AIDS, the AIDS virus eradicates the CD4 cells. So if you don't have them, you can't mount an immune response. All right, more on those later. The TC, C is for cytotoxic. Sometimes they used to be called the T killer cells because they can attack a virus-infected cell one-on-one. They don't secrete antibodies to do it. They do it themselves. And they're also called the Cd8 cells. The Treg, regulatory, these are like suppressor cells. Sometimes called, well, they have a CD25 molecule. I won't say too much about them other than, well, they're like the brake. They stop the whole thing. Hence, suppressor, brake, stop. If you have a problem with your this subpopulation, people usually have problems with allergies, autoimmune responses, because you can't turn the thing off. So they're the brake. And then you have memory helper T cells. So these helper Ts can be memory. The CD4s and CD8s both can be memory, so I'll just kind of do this. Can be, quote unquote, a memory cell. Memory meaning you've reacted to the bug already, you mount a stronger, faster response, just like for the memory B cells. So let's talk about the B cells first. Again, in both cases, B or T, it requires exposure. In the case of the B cells, the antibodies have specific binding sites for the antigenic determinants on the bug. So I already wrote that it requires exposure, but that is the provocation of the immune response. And so if you present more different shapes of antigenic determinants, the more chances you have a clone that can mount a response. The more the better. In this case, how many clones were activated? 3. 1 for each antigenic determinant. All right, so the B cells, I already said, they secrete antibodies, and I already defined the subpopulation. You should appreciate the great diversity of clones you have. There's something called somatic recombination, where you just shuffle the genes that produce these different antibody clones. And the reshuffling results in a remarkable diversity of antibody, of different kinds of antibodies, 10 to the 11th power. That's a huge number. If you look at the immunoglobulin structure, I just want you to know that the 4th region is the specific region. So that when you have a primary response, at the top of the screen there you have 3 clones. All right, 3 clones. And the middle clone is specific for that bug. So you note that the antigens are binding the membrane-bound antibodies that act as the receptor, and that clone is proliferated. And then you get your plasma cells, and some persist as memory B cells. But what will get you better for this first infection are these antibodies from the plasma cells. But let's say you get well, and then maybe years later, you get that same bug again. Not likely, but it's possible, because these things mutate. But the point is, the same bug, memory cell, triggers a faster, stronger response, and you probably don't get sick. So the important thing that you can measure in the lab are the antibodies. We call it antibody titer. And the initial response is slower and weaker. The second response is faster and stronger. But if you get infected by another bug, well, it's like the primary response for A. Okay? So there's a difference between primary and secondary response, depending on which exposure is it. Is it first exposure? Is it second exposure? So what we expect students to know for B cells, there's a difference between the primary response and secondary response. So the first response is weaker, slower, I guess, so the right words, stronger, faster. Remember, these are the ones that are secreting the antibodies, the ABs. So that's pretty easy to understand. So on this slide, it shows you how the antibodies are the accessories to murder, but they're not the murderers. So if you look at that middle row here, this is how they just kind of bind things up for you. They either fix complement, they coat, they fix, they precipitate, they bind these soluble antigens, they can agglutinate red blood cells that are non-self, they neutralize other bacterial endotoxins, viruses. But what those do is, what this does is, it leads to lysis. That's the destruction. It leads to inflammation and phagocytosis. That's the destruction. Okay. Now, there's different ways we can acquire immunity from B cells. You can either do it actively or passively. So we call this acquired immunity. That's a good thing. You acquire immunity. You're just building your army of lymphocytes and antibodies. So you have active and passive immunity. There's two categories of acquired immunity. If immunity is actively acquired, what that basically means is you're getting the memory cell. Passive immunity means you receive the antibodies. Passively acquired, just get ABs. Not the cell, just the preformed antibodies. Okay, so okay, let me ask you this. Which one's better? Getting the B cell or getting the antibodies? Well, what do you think lasts longer? The cell. They last indefinitely. The antibodies don't last very long at all. Maybe hours, maybe days at most. It depends. Well, there's different subcategories of active and passive, and this kind of shows you that. They call this the humoral immunity, depending on how you get the cells or the antibodies. So for active, there's a naturally acquired, artificially acquired category. So for the naturally, well, that's easy to understand. Just you get sick. Someone coughed on you. You know, you coughed on someone. Maybe you're patient zero. You got someone else sick. Someone got you sick. It all happens both ways. Okay. But when you get sick and you survive, it doesn't kill you, make you stronger because you get the memory cell. You're not getting that bug again. So artificially acquired, that's get vaccinated. You intentionally go get the flu shot right around now, actually. Flu season's typically January and February. And so, but that's the amount of time you need to react to it, okay, to get the antibodies up, so that come flu season, you're pretty well protected. Even if you do get sick, it should be far less than if you didn't get it. So the vaccination's supposed to help. For the passive, just get the ABs. You usually don't do that unless you're pretty close to death. Okay, so in terms of, well, let's do the artificially first. Artificially. A good example is the snake bit patient, close to death. And you're in dire need. So what you can do in preparation in your clinic, say in Arizona or something where they got snakes, that's a common thing. Take a small animal, inject it with a little bit of the snake venom. Inject small animal with a bit of snake venom. You should choose the one where you see the most cases of snake bit patients. So inject animal with snake venom. And then, well, not enough to kill the animal, but what you do is you harvest the anti-snake sera from that animal. Harvest anti-snake sera. Okay, and that's what you give to the snake bit patient, the anti-snake sera, to help save them. Because you assume the animal, which mounted an immune response, there should be enough there to help your human patient. All right, so the way you can naturally acquire preformed antibodies is from mom to baby. Naturally. There are subclasses of antibodies. I've been calling them ABs, but frequently you'll see them are called IG, immunoglobulin, and there are different subclasses of the IGs. For example, IgG This is like about 80% of them. IgGs are ones that can cross the placenta from mom to baby. IgGs cross placenta. And from mom to baby, even after baby is born, those can persist for maybe 8 to 10 months. Then the other thing to consider is mom nursing. I'll put a question mark, because not everyone does, for a variety of reasons. But breast milk, if you are nursing, contains the subclass IgA antibody, so the baby will get those immunoglobulins from the breast milk. And, well, if you nurse for 1 to 2 years, that's another 1 to 2 years of protection. Okay. Okay. We'll take a break. When we come back, we'll get into the T cells. One thing before you break, come back at 12:30. When we come back, we'll finish these slides. And then once I finish with these slides, we're going to go right into the exam. There will be no break, so just kind of know that before this break. This is the only break I'm going to give you.